that question deserves a nuanced answer because gliomas are not one type of disease. There's different types of gliomas. And you can broadly separate these by age, so the age of diagnosis. So for example, oligodendrogliomas often occur in younger people in their 30s or 40s. Astrocytomas broadly occur in 40s and 50s, and glioblastomas later in life, 50s to 60s. And that also relates to outcome of these patients. So oligodendrogliomas have very favorable outcome. These patients live 10, 15 years, sometimes 20 years. Whereas a glioblastoma, the older patients, they have a dismal prognosis, only 15 months or 18 months. However, if you look at years lost due to cancer, because the oligos occur earlier in life, actually the number of years lost due to cancer is as great, if not greater, than the glioblastoma. So they're universally very bad diseases. It's hard to compare them to other types of cancer because you have similar spectra of outcomes and nuanced subtypes within breast cancer and within liver cancer and so on and so forth.
I'm not an expert, but it looks like to me that when you have a cancer and you are younger, it might be worse than when you are older, but it doesn't occur in gliomas. Is it different from other types of cancer in general?
So yeah, most cancers affect more older patients. So in that sense, gliomas, especially the oligos and the astrocytomas, they tend to occur earlier in life. But then if you think about familial breast cancer, people with an hereditary defect that may predispose to breast cancer, those also tend to occur early 30s, 40s. And if you look at the numbers, there's probably more of those types of patients than, fortunately, or because they're better treatable, fortunately, than gliomas. Having said that, any patient with a cancer that's in their 30s is going to have a huge effect, both in terms of quality of life due to the treatments. It's going to affect a lot of, that's of course a time in your life that you tend to have a family. You tend to be building your career. So it's going to have a tremendous impact, whatever the cancer type is.
What about the perspectives of treatment between gliomas and other cancers?
So for treatment, the most commonly used treatments are chemotherapy and radiotherapy, so radiation. So for the types of glioma that we consider to be less aggressive, for example, oligodendroglioma, excuse me, there's no standard of care there. There's some studies that suggest that chemotherapy may be beneficial. But these studies take a very long time because these patients survive relatively long, so it takes a long time before we can actually measure a difference. But chemotherapy is the most commonly used type of treatment for lower grade patients. For glioblastoma, we have a standard of care which consists of chemotherapy and radiotherapy at the same time. And this improves survival, but despite that, survival is still very poor.
And surgery is also an option.
Yeah, surgery is of course the standard for every glioma.
So you have to first be on a surgery, then you treat with the other treatments, right?
That's right. There's no neoadjuvant treatment, so there's no treatment up front in general. This surgery is the basis. You debulk the tumor, as we call that. And after that, the oncologist may decide that chemotherapy is the best choice, or in some cases, radiotherapy. And in glioblastoma, it's both.
You were the first one to make a specific about astrocytomas and oligodendrogliomas. What are the difference between them? Is it just a morphological difference, or they are really different?
So there's a morphological difference, clearly. So that means that when a pathologist looks at these tumors under a microscope, you can distinguish the different cell types. If you analyze these tumors using molecular profiling, you can also clearly separate them. In fact, we can actually do a much more precise separation. There are some tumors that are called oligoastrocytomas, in which case the pathologist sees both oligo-like cells and astro-like cells. So he or she is not able to make a distinction But however, molecularly, the distinction is very clear, and we don't actually consider the category of oligoastrocytoma anymore.
So these differences are also different between these low-grade gliomas and glioblastomas? Are these different?
Exactly. Well, in glioblastoma, you add a component of tumor-associated normal cells. So you tend to see immune cells that associate with the tumor at a much greater frequency than with the lower grade astrocytomas and the oligodendrogliomas. And you tend to see a much more necrosis. So cells that affect are dying in the center of the tumor, which is a bit paradoxical.
Oh, sorry.
Which is a bit paradoxical that there's tumor cells that are dying in this tumor type that is very aggressive, but that's what we see.
And what about the molecular analysis? Does it also offer different perspectives from each one of them?
Yeah, the most a huge separation is the presence of a mutation in a gene called isocitrate dehydrogenase. It's IDH in short. When a glioma is determined to have a mutation in IDH, this puts that glioma in an entirely different category from the IDH wild type tumors. So IDH wild type is when there's no mutation, when it's the same as the germline. Those tumors, those gliomas that are IDH wild type are in majority glioblastomas, and those are the tumors that do the worst. Those patients with those tumors do the worst, and they deserve the most intense treatment that they can bear, in fact.
Now, I would like to compare the importance of the molecular findings and the importance of epigenetic studies. So, what did scientists know before you have this molecular analysis, and how does this analysis can complement this knowledge?
So, before we had these types of technologies available, in hindsight, it's easy to say we knew very little. We knew about some point mutations, such as in genes called P53 and EGFR. We knew that those were important. But since we've been able to do these molecular profiling studies, we've amended tremendous insight into how to best classify gliomas, and the epigenetics is a huge component of that. So the gene that I just mentioned, the IDH gene, was discovered only in 2008. So, within the last 10 years. And that by itself is in a way has been a breakthrough, because we've seen so many differences that are associated with whether a glioma has a mutation in that gene or whether it doesn't. And that also follows along the lines of the epigenomic studies. So a glioma that carries a mutation in IDH has a very different epigenetic profile than a glioma that does not have a mutation in IDH.
Does do these differences influence in any way the way we treat and diagnose patients?
So they certainly do. For example, previously when we did, we made a diagnosis on the basis of pathology, on the basis of a glass slide and looking at that glass slide under a microscope, we could classify a patient, a tumor as a low grade, as a high grade, as an astrocytoma, as an oligodendroglioma. However, what we discovered in this study, and also follows up on previous discoveries, is that there are low grade tumors that, despite being low grade, they don't have a mutation in IDH. And the absence of the mutation in IDH immediately puts that tumor in the worst category, unfortunately, for those patients. And being in that worst category means those patients really should get the standard of care for glioblastoma. So that has a huge impact for those patients. On the other side, we also have found that cases that carry the mutation in IDH, and subsets of those do so well that perhaps they could do without some type of therapy, which is not entirely uncommon. There, for low-grade tumors, it's not entirely uncommon to not follow up with treatment, aside from surgery, of course. So I think on both ends of the spectrum, I think our study provides insights that are clinically relevant.
So what would be the main discoveries you can explain to us? Not explain, just say to us.
Okay, so the main discoveries in our study would be, so we knew previously, before our study, we already knew about mutations in IDH. And we knew about a second category on top of the IDH that we call 1p19q codeletion. So we knew about these, and we knew that those two factors are tremendously important for classification of gliomas. So previously we determined that there were, across the board of adult diffuse gliomas, that there were three groups. Cases that carried IDH mutation. On top of that, they had an 1p19q codeletion. The second category was IDH mutant, but in absence of that codeletion. And the third category was IDH wild type, so no mutations in IDH. We expand that classification. We don't recognize just these three subtypes. We actually now think that there are, across the board, 7 groups. Within that group of IDH wild type gliomas, there's actually a subset that does relatively well, whose survival is much better than of other IDH wild type tumors.
What is the name of it?
Sorry?
What's the name of this group?
We call them the pilocytic astrocytoma-like group, because pilocytic astrocytoma is a type of glioma that typically occurs in pediatric patients, in children. And this subgroup, although these are adult patients, carry many characteristics that we typically associate with pilocytic astrocytoma. However, that diagnosis is made on the basis of pathology. And when we re-reviewed these specific adult gliomas, they were definitely gliomas. They were not misdiagnosed. So that group, PA-like or pilocytic astrocytoma-like, could be treated differently. It's a little too early to say that because our observations were made on the basis of a relatively small number of cases, although extremely robust. There's no doubt about the reality of this subgroup. It's a little too early to really say what that means clinically, but we should distinguish this subgroup. On the other side of the spectrum, within the category of IDH mutant tumors, we discovered the reverse. So remember, the IDH mutant tumors do relatively well. However, we discovered a pocket of patients whose prognosis is dismal, almost as poor as glioblastomas, but we can really predict that up front. And these cases those will be candidates for very intense treatment, potentially the standard of care for glioblastoma again.
So can you describe shortly the 7 subtypes you described in the paper?
So the 7 subtypes, wait.
I have mine here.
I'm just going to make sure that I don't skip any. Okay. So we have, within the 2 broader categories of IDH mutant and IDH wild type. So within IDH, let me start with the IDH mutants. Okay. We think that there are 3 subtypes of IDH mutant glioma. The first is the codel group that I just described. So these are cases that have an IDH mutation, and on top of that, they carry a 1p19q codeletion. This is a favorable outcome group, even though these patients are typically diagnosed when they're younger, so maybe early 30s. But their outcome pattern is favorable. They may live 10, 15 years. I'm not saying that's good enough. I'm just saying that's favorable relative to other subtypes. Then we have a second group that we named GSIMP high. This group actually also has very favorable outcome. Even though these patients, on average, their age at diagnosis is a little higher, they may be late 30s or 40s. Despite that, their survival is as good as the codels, so 10, 15 years. And then there's this third group that we labeled GSIMP low, and that's the subgroup that does poorly. So they are not recognizable by just looking at 1 or 2 genes. You really would need to do generate an epigenetic profile to recognize this subgroup. But these are patients whose tumor is aggressive, and they recur within a much shorter time than other types of IDH mutant glioma. And these are candidates for more intense therapy. So those are the 3 groups of IDH mutant tumors. Then we have the second larger group of IDH wild type gliomas. And within this group, we have 4 subtypes. We have groups that we labeled classic-like and mesenchymal-like on the basis of their molecular features. These are the classic-like and the mesenchymal-like are both typical glioblastomas. They have high levels of necrosis, they are aggressive tumors, they deserve the standard of care for glioblastoma all the way, no doubt about that.
And what
Then,
do differ one from the other?
so the third group that we recognized, we labeled LGM6 GBM. These are not quite the traditional glioblastomas, but their clinical pattern is exactly the same. So they're aggressive tumors, they deserve the standard of care for glioblastoma. There are some nuanced differences that we don't quite understand. And then there's the fourth group, the group that we labeled pilocytic astrocytoma-like. These are lower grade tumors. They're IDH wild type, but they're lower grade tumors. They carry features of pilocytic astrocytoma, a pediatric type of glioma, and their outcome is relatively favorable. So 5 years, 6 years on average, which is favorable if you compare to other types of glioblastoma.
I'll try to provoke you a little bit, but this classification, the physiologic and the pathologic analysis would be like they are, for instance, PA-like or LGG or low-grade gliomas, they would be in the most favorable outcome group. And by the other hand, the G-simp low are prevalently GBM, right? Glioblastomas, yes. Mm-hmm. And they would be in the worst case scenario. So isn't this classification making more confusion about it or not?
I don't think so. Actually, I think it clarifies many things. So when you say pilocytic astrocytoma-like, those are lower grade tumors that are IDH wild type. However, only roughly half of the lower grade tumors that are IDH wild type fell into this PA-like group.
Oh.
So not all, there's a, within that category of IDH wild type lower grade tumors, mm-hmm. It's really critical that this distinction is being made. Yeah. At the other side, the G-simp lows, it's exactly the same. So the G-simp lows are roughly half of these are grade 4 tumors or glioblastomas. However, not all glioblastoma IDH mutant tumors fell into this G-simp low group. So again, it's critical that this distinction is being made, because you may be classified as a grade 4 glioblastoma, you may be IDH mutated, which determines that your prognosis is relatively favorable. However, based on this epigenetic classification, it could be actually very favorable, or actually it could turn around and could become very unfavorable.
Perfect. You said just one thing that that reminds me that you said the epigenetic to low-grade gliomas, it would be no, to G-simp low, you have to use an epigenetic classification. But back in the paper I read it, it was just different from IDH level, not IDH, methylation level.
Yeah, that's right. So
this is the epigenetic classification, the methylation level?
That's right. Those
okay.
are the same thing.
Great.
Those are the same thing.
How do you think it can be translated to the clinic, to the daily clinic? Do you think this study can become a way of diagnosing real patients?
Yes, I believe very strongly that this should be the way that patients are being diagnosed. So, of course, we need to distinguish that we need to acknowledge that this is an assay that requires some technology, some infrastructure that needs to be in place, and that's not going to be feasible immediately for everybody. And most likely, the most well-known cancer centers in the U.S., in Europe, Japan, Australia, those are most likely to be able to offer these types of technologies first. That's just the reality of this. There's a center in Germany, in Heidelberg, where they've adopted this technology, the methylation profiling technology, to classify all their brain tumor cases, not just gliomas, but all brain tumor cases, pediatric, but also all adult types of brain tumors, meningiomas, epidemiomas, and so on and so forth. There's a number of other institutions in North America that are investigating whether they could implement something similar, including my own center, MD Anderson. We are actively trying to figure out how can we make this part of our clinical practice. An institute in Toronto is quite advanced in this as well. I don't think they've actually rolled it out just yet, but they're definitely trying to make this happen. And I think we are setting the trend, and I think this will be followed once we are showing that this is actually improves patients' outcomes. I think others will follow.
So the only machine technology you have to have is the one that separates from IDH from methylation profiles. It's just this one you need to have to make a better diagnosis.
Yeah, and on top of that, for that one group, you need to have a pathologist determine the grade. Yes. But that's right. It could be a single assay that is used to determine the classification. And again, in Heidelberg at the DKFZ in Heidelberg, this is what they're doing.
Do you think there are differences that can be that are shown from the way they? Sorry. Do you think from when they started doing this and now they can already see differences between the treatments of the patients and the prognosis? Is it better or worse? Do you know they have these results?
So what they're doing at Heidelberg right now is trying to determine the classical diagnosis using methylation profiling. So they're not trying to discover what is the optimal way to classify gliomas. They're trying to figure out, can we predict when we call something an astrocytoma, can we predict that it's an astrocytoma? So it's a slightly different approach.
But will they be able to use these 7 subtypes that you propose in this paper after you publish this paper?
Absolutely, they would be able to use those immediately. And I think once our paper is published, I'm hoping that I can also get the support to implement the same 7 subtypes in my own institution, that we can lead the way in how glioma should be classified. And then, of course, we also hope that others will follow. And I have no stock in Illumina or any other methylation profiling companies. This is because I believe this is the way glioma should be classified.
So in general, what do you think there are the outcomes, the possible outcomes from this study?
So for the small groups of cases where we initially thought these are IDH mutants, so they should do well, but they don't do well, and we kind of recognize when they are not going to do well. Those patients hopefully will be recognized as such and thereby get treatment that is more aggressive, and hopefully their life will be prolonged. On the other side of the spectrum, those pilocytic astrocytoma groups, they may benefit from, I don't think they should be taken off therapy in any way. Their outcome is not good enough for that, but they have specific alterations. They may be more susceptible to BRAF inhibition, for example, and there may be opportunities there. I think that will be worth a clinical trial.
Just to finish, I would like to explain some aspects. First of all, what is this IDH mutation? Where is the gene mutated?
So the gene, it's always the same. It's a so-called hotspot mutation, which means that in this gene, it's always exactly the same base, or roughly maybe with 1 or 2 bases difference, but in general, it's the same base that is mutated, it's the same nucleotide. It's a gene with multiple functions. The most prominent function is in the processing of metabolites. It's part of the energy producing machine in the cell. In fact, there's been studies that show when a cell carries this mutation, you see an increase in particular metabolites and a decrease in other metabolites. There was an exciting publication last week that suggested that this actually provides a vulnerability in these gliomas, that they may actually be treated not on the basis of their mutation, but on the basis of the high level of that specific metabolite. And that taking away that metabolite actually makes the cells vulnerable to therapy. So there's advances in that aspect as well.
What is this metabolite?
I would have to look up the exact word. It's in short, it's 2-HG.
2-HG.
I think it's 2-hydroxyglutamate.
Okay. No, just for me to know. It's not on record on the interview.
And there was if you want to look up, there's a publication in Cancer Cell last week that looked specifically at this, and it was they use NAD+ inhibitors, which affect specifically the metabolism, not so much the mutation.
When you say the metabolism, you're talking about the mitochondria?
Amongst others, I mean, it's the way the cell produces and the energy needed to keep itself going.
But inside the mitochondria?
Yeah, most of these processes occur in the mitochondria, but not all. There's processes, mostly the I have to dig back in my biochemistry course now, but I think the oxidative metabolism occurs in the mitochondria. But there's also non-oxidative processes, and I think those may also occur in the cytosol and not so much in the mitochondria.
No, okay. And what about the 1p19q deletion? What is it exactly?
So this is a very broad event, whereas the IDH mutation is a one nucleotide that gets changed. So only one position in an entire genome of 3 billion bases, one position gets changed. And that predisposes you.
Sorry, where is it exactly for me to draw? For instance, I would draw a gene and say, well, it's in the beginning, the middle, and do you know where is it? What it is exactly?
It's not in the beginning. It's in the middle. It's not a huge gene. It's roughly in the middle. Maybe in the first 1/3, but then around 30% or 40%, I think, of the gene.
Perfect. I'm sorry. Let's go back to the 1p19q.
So 1p19q is a big event, whereas the IDH mutation is a single nucleotide that gets changed. In the 1p19q event, what happens is that part of chromosome 1 gets fused to part of chromosome 19, and the cell is unable to sustain that fusion, so it deletes the entire fusion. Or cells that delete that fusion are able to survive, and cells that are not able to delete the fusion don't survive. So you get a fusion of 1p and 19q, then you lose the fused 1p19q, and therefore it actually shows up as a deletion. So now you only have one copy of 1p and you only have one copy of 19q.
Oh, instead of
two copies, which would be the standard, yeah.
I thought all chromosomes 1 would lose one leg and all the chromosomes 19 would lose another. It's so just one that occurred the fusion.
Yeah, it's called a heterozygous deletion, which means you maintain one of the arms of chromosome 1. Probably because you can't lose an entire chromosome arm and still have the cell being able to be alive.
Great. And I also have a curiosity to ask, but how could you coordinate a group of people that were in several places at the same time, needing the results from one another and having to talk about them all the time? How is it to coordinate such a great group?
So yeah, this is very much like herding cats. So in this instance, we had an immediate core group of investigators in Brazil, in Qatar, in Italy, in New York, and then ourselves in Houston. This was the core group. But then beyond that, we had a much larger group with people all over the country. And by the country, I mean the US, and beyond the US in other also international partners. And the way we ran this study is by having very frequent teleconferences. So we would have a fixed time point in the week where we would have a teleconference. Once we moved closer to a manuscript, we focused with the core group on very frequent teleconferences, exchanging files through the web, like using Dropbox, using Google Drive, using Google Documents, and just trying to be in touch frequently. It was occasionally a painful process, but I think the group of people that we worked with was extremely collaborative and extremely willing to move this forward, and thereby I think we came out with a great set of results that matured into a great manuscript.
And why was it necessary to have all these groups together?
So each of the groups brings with them a particular expertise. So for example, the group in Columbia is a senior investigator with very much knowledge about gliomas and a vision on where the field is going, and brought with him also postdoctoral fellows that could do computational analysis, the part of the sort that is needed for these types of studies. The person working from Qatar, for example, was very much leading the mutation analysis, so looking at which bases change and so on and so forth. The group in Brazil was leading most of the epigenetic analyses. Right? So they were driving that aspect. And my group here in Houston, I think we oversaw the whole project and filled in on many of the gaps. For example, a lot of the clinical analysis, the survival analysis, was done here. We looked at the telomeres, which is another aspect of the study that we haven't specifically discussed now. But altogether, it was extremely synergistic, and I think the sum of this group is much greater than its individual parts.
And do you think that, talking about the telomeres, you just remember me, can you, the analyses were really broad, right? You look at several aspects, but which aspects do you think are really relevant for this classification?
So the classification starts on the basis of epigenetics. Epigenetic profiles they reflect biology of the cell. That biology may be related to the type of cells in which the cancer originates. For example, the cell of origin, or it may reflect the effects of very important, very critical driving alterations that occurred in those cells.
What I meant, I'm sorry, I don't think I was clear. I meant that when you have all these characteristics, the ATRX, the IDH mutation, the codeletion, and everything, is it important to study all of them, but even that some are not relevant for this classification?
Mm-hmm. So again, there's two answers, because there's the answer of what is immediately important to the patient, and to the patient, you can generate the epigenetic profile, you can classify them according to our 7 groups, and that's all they need to know. That's all the physician, the oncologist needs to know to determine what is the optimal way to move forward in treating this patient. So that's one aspect. Then there's the aspect of research, and for this, I think they're actually all important. It's important to know what the TERT status is. It's important to know what the epigenetic profile is like. It's important to know what the ATRX is like. If you really want to understand how one specific glioma is moving forward, how it's maintaining itself, how it's proliferating, you need to know all these different aspects. You need to have that holistic view of the glioma system in order to really understand what is happening in my study.
And really understand, you mean that you can also look at some targets that maybe will become a treatment, or?
Exactly. I mean, if you want to study what are vulnerabilities in a particular cell, it's important that you realize that a cell that carries a TERT mutation is different from a cell that does not carry a TERT mutation. It's important to realize that a cell with long telomeres may be affected by different treatments than a cell that has short telomeres. So in a perfect world where resources are not an issue, you need to know all these different aspects to really understand what you're looking at. Cancer is a complex system, and it's not getting any easier, but we're trying to create some order in chaos, I guess.
That's a great definition. Thank you very much, Dr. Roel.